promised, this is the second installment of my discussion today, and again, focusing on research components. Here we go. So again, I've modified the title a bit. Thank you to Dr. Bradley for setting this up, right? So we think about, taking this to the next level. We think about dystonia, blepharospasm being a subtype of dystonia relating to dysfunction of the CNS, the central nervous system, or the brain. And specifically, we're starting to think about this as dysfunction of networks and how regions of the brain communicate. And so I'm going to talk about these more advanced research priorities. I'm really going to specific as an expert in neuroimaging on applying multimodal neuroimaging methods to discover both molecular and network dysfunction in dystonia. And my disclosures have not changed with the exception of a free lunch today from the BEBRF, so I appreciate that. Thank you. So this talk, we're going to talk about the research priorities in dystonia. We're going to highlight the second part of those priorities that I mentioned before. We're going to highlight blepharospasm genetic research. I'm not an expert in genetics research, but I do want to bring up that component because that is a priority of which there is some progress. And then I'm going to really focus in on neuroimaging tools, the relevance to understanding brain dysfunction in dystonia, and we're going to highlight the ongoing research efforts to determine common molecular and circuit level or network dysfunction in patients with dystonia. And so this is the list I showed earlier of research priorities that was defined at the NIH level. And we focused earlier on the clinical characterization and the natural history. Now we're going to briefly talk about the genetics and then focus on neuroimaging, molecular, biochemical components, and how we're applying advanced technology to learn more about these diseases. This is a slide that really just shows that genetics represents a small piece of the pie in dystonia. This is a identified gene variants in a population of isolated dystonia. Isolated being dystonia is the only problem. And as you can see, only about 4% of that population have an identified gene that is known or thought to contribute to the dystonia. So it's a very small population, a very low probability that known genes contribute. The emphasis there is known, and that we're learning more every day. We're learning about more genes that contribute and how they contribute. And then this wheel, I recognize that folks can't see this and this doesn't project very well, but this wheel here is the isolated dystonia components, and the point is that there are multiple genes, 1, 2, 3, 4, 5, 6, 7, 8 as I go around that wheel, that have been associated with the focal isolated dystonia, not specifically blepharospasm at a high level. So more to be determined there. Those with blepharospasm who have undergone screening, particularly those with a known family history, there have been 16 plus identified gene variants in 4 relatively large studies. So the authors' names, Tien, Hammer, Tang, Selman, and the N is the number of patients with blepharospasm who have been included in that study. So not a lot of patients with genetics known, and then certainly I'm not going to test you on the genes that I've listed here. All those funny consonants and numbers are the various genes that have been identified. I do highlight REEP4 because there's a relative convergence of that gene in blepharospasm, but more to be determined. The other consideration is that these genes, these identified genes, don't just cause blepharospasm. This, again, I know this isn't projecting well, but this first column here is the incident cases of blepharospasm specific compared to other focal dystonias. And that's for one gene here, another gene here, another gene here, another gene here. And so the point of that is these genes are not specific to blepharospasm. They're also contributing to other forms of dystonia. So we're just tipping the tip of the iceberg with regard to genetics. We don't have a clear association yet, but this is something that shows a lot of promise in the field, and certainly is the holy grail that I think Dr. Holtz or someone else mentioned earlier today as far as being able to potentially modify that and really cure dystonia if we can identify that. But shifting gears relatively dramatically here, I want to kind of pose the principles of neuroimaging, right? So neuroimaging is where we take pictures of the brain. And we take pictures of the brain to look at structure, right? Are all the parts there? And we can take now pictures of the brain to look at function. Are the regions of the brain communicating well? Are they communicating like we anticipate or not? And there is increasing evidence that regions of the brain do not communicate well in dystonia, and that lack of communication is contributing to this. Neuroimaging is a growing field. all hospitals, all academic centers have access to MRI machines, PET machines. These are products that we use to make diagnoses all the time. And the idea in researching dystonia is that we want to use these tools to direct the spotlight on relevant brain regions, right? So if I can determine, taking a picture of the brain, that a particular brain region is affected, then we can really share with our colleagues that, and they can start shining the microscope on that, literally, right? They can look at the molecules, the neurotransmitters, the genes in those regions, and try to better understand that. And so I'm going to kind of guide you down a path in which we've successfully done that, thinking about the components of neuroimaging that are available. And so this path starts with structural neuroimaging. In dystonia, it's been mentioned, when we take pictures of the brain in people with isolated blepharospasm or other forms of dystonia, nearly all the time the brain is completely normal on the conventional scans that we take, CT, MRI, those types of things. There are rare instances where someone may have a known pathology, a stroke for instance, and shortly after they develop that stroke, they develop dystonia, symptoms of dystonia. And so these pictures here are to kind of convey, again, what we know as far as the regions being affected. So on the left here, this is just orientation, right? So this is a human head, the nose, they're facing out towards us, and we slice the brain like this, and we pull the top off, and we can look down on the brain. And so that's what this picture here shows. This is a CT image, and this is a normal brain. We see some tissue, and then we see kind of some dark areas in here, which is fluid that surrounds the brain. And so we see contrast in that brain, and you're just going to have to trust me on that if you're not a neuroimaging expert. But on this image here, so this was the work of Dr. Marsden in the 1980s, actually, where he recognized this, that patients would have strokes, they would develop dystonia, and he looked across all those patients that he had access to and noticed that the most common region that was damaged was this region here. And this is that basal ganglia that keeps coming up, right? So this region of the brain commonly, when injured, results in dystonia. And so that's a clue as to what might be causing dystonia. And again, it's shining that spotlight in that region. So we've spent a lot of our time over the last 20, 30 years really focusing on this particular region and trying to tease it apart, come up with, understand the chemical mechanisms and the components that we can treat there. There has been a relatively recent shift in that dogma as we have increased technology, as we have increased access to patients with dystonia and recognition of it. Again, busy slide. The point of this slide is that following stroke and a reassessment of this by a group in Cincinnati in 2018 demonstrate that not only the stroke of the basal ganglia contribute to dystonia, but so does stroke of the motor cortex, so does stroke of the thalamus, so does stroke of the cerebellum. These are all different brain regions that are involved in motor control, but in very different regions. And so this suggests that historically, while the basal ganglia is important, and I'll go back to that this dystonia likely represents a dysfunction of how these regions that control motion and motor control are affected and not communicating well. And so this is the concept of dystonia reflecting a network disorder in the brain. And so, again, in development of technology, we now have tools where we can specifically look at the networks in dystonia. And this is specifically called functional connectivity MRI. Functional connectivity MRI, so this is kind of different from standard clinical MRI where we just look at the structure of the brain, right? We take pictures, there's lots of physical properties that allow us to see the structure of the brain. Functional connectivity MRI takes it to another level. We can look at the blood oxygen level and how that changes based on essentially what is happening in the brain, what the activity is, what the neurons, the cells of the brain are doing. And again, complex principles, but the idea being that we can measure neuronal activity or the product of neuronal activity in one region of the brain, look at how the blood flow changes in that particular part of the brain, and we can do that in ways that we can control. For instance, we can put someone in an MRI scanner and we can have them shine a light in their eyes, have them open their eyes, and then have them close their eyes. We can compare the differences in the brain in those different states, and we observe that the occipital lobe, the back of the brain, often lights up, right? We see activity in that when patients are looking at light, looking at things. And so that gives us a clue that that occipital lobe is involved in the visual processing, right? Dystonia is a little bit trickier. There's no action per se that causes dystonia, or at least if there is, then we start to run into confounds of interpretation. I'll talk about that in a little bit. But to advance this technology a little bit more, take this yet another level higher, we have this concept of resting state MRI. And this concept is that if one region of the brain, when people lie in this scanner, the MRI scanner, the blood oxygen levels fluctuate naturally. And they do so in this kind of rhythmic way that we see here. And so I am interested in the basal ganglia. I've drawn a region in this. And over time, I can look at the blood flow, the oxygen levels in that region, and see it fluctuate over time. That's not particularly interesting in and of itself, but what gets really interesting is that regions that are functionally comparable, they serve the same function, the blood oxygen levels tend to fluctuate at the same level. They correlate very highly, and so that's demonstrated here. As opposed to regions that don't have similar functions, they occur more randomly in association with each other. So this tool allows us to look at the network, the brain network, and how it functions in individuals. This science has blasted off in the last decade. Everyone has an MRI scanner. Everyone has access to this. This is really exciting science. The problem is, in the wrong hands, it produces very poorly reliable data. And this is a great example of this. I don't know how many people. So this is an MRI image, a picture of structurally of some object or animal. I don't know if anybody recognizes that. It's a salmon. Yeah, good. And these researchers put this salmon in this MRI, and they detected statistically in this powerful method this little red spot, which is right in the brain area of the salmon. The problem is the salmon's dead. It doesn't have brain activity, right? So this was a big hit in the field with respect to how we're approaching this and the statistical noise. And so this demonstrates that we really have to pay very close attention to this science and what we're doing to study these networks. And this is a caveat that we take very seriously in the field now and have applied different methods, very sophisticated methods, to make sure statistically that we're actually measuring the signal, not just noise, right? In dystonia, we've set out to use this tool. We want to process high quality data in a very sophisticated statistical method across large populations of awake resting participants with various forms of dystonia. And we want to compare those to healthy controls, people who don't have dystonia. And we want to determine what is the difference in the brain network in those who have dystonia and those who don't. Sorry, I got a flash there I got to get rid of. And so we did this at Washington University in our group. We compared, and another component of this is we wanted to find common network dysfunction across dystonia subtypes. Again, this would be very powerful because we can shine that spotlight not only in a small subgroup, but apply methods across these dystonia subtypes. Is there a common circuit level brain dysfunction? And so to answer that question, we included patients with laryngeal dystonia, with cranial dystonia, blepharospasm, upper limb dystonia, and cervical dystonia. We scanned all of them at rest, and we pulled their data together. And we, in particular, looked at this region of the brain, the basal ganglia. This is a zoomed in segment of the basal ganglia, for which Dr. Simonian, who Dr. Bradley had mentioned earlier, had studied. She had looked at neurotransmitter levels in that region, noticed that they were reduced. When we apply that particular region, we draw a seed on that in our MRI data set. Sure enough, we demonstrate that there is decreased, weaker brain functional connectivity or brain networks within the basal ganglia structures of the brain. And so this helps to support that notion that there's network dysfunction in dystonia, and solidifies that notion that the basal ganglia is a large part of that component. These concepts are particularly relevant for therapeutic potential. Neuromodulation. I've had a few questions and we've heard a little bit about deep brain stimulation surgery. Deep brain stimulation surgery is essentially a pacemaker for the brain. We're inserting a wire into the brain, and we're disrupting how information flows through these networks. And so the thought being that now we've solidified this region, basal ganglia. We know that DBS to that region helps. This notion gets a little bit more complex because we have done this at a group level, right? So I don't have a slide to represent this, but in somebody who has blepharospasm, the region there's a principle of somatotopy. The brain reflects regions based on the body that it controls. The voice, there's a brain region that controls the voice. There's a brain region that controls the eyes and the blinking. There's a brain region that controls the neck. So we are now taking this to a new level to start looking at these brain networks in individual patients. Can we guide this wire in DBS or this stimulator in TMS to be directed at that specific body region in an individual, as opposed to the average across individuals, which may very well differ? We're also going to attempt to link this network dysfunction back to biochemical dysfunction, right? There's a fair number in the audience who are reluctant to undergo brain surgery, to have wires placed into their brain. So how can we learn from this dysfunction, and for those that don't want wires in their brain, to manipulate the brain, to apply potentially medications that would, again, correct the biochemical dysfunction? Biochemical in this sense, neurotransmitters, signals that traverse the brain to allow the brain to communicate within itself, but also to send signals to the muscles of the body, and that contributing to the dystonia. And so this is what we talked about earlier. We categorize dystonia. We look at the clinical phenotypes, or what we observe on the patient. We've now demonstrated that functional connectivity MRI, looking at the brain circuits, the abnormal networks, abnormal networks do contribute to various forms of dystonia. Let's take that a step back. Let's look at the biochemistry. Let's understand what's going on at that molecular level in this particular region that's affected. Again, way complicated slide. The point here is that we have a brake pedal and we have a gas pedal in the brain. The basal ganglia essentially controls how much pressure is on the brake or how much pressure is on the gas. And that is largely determined by a particular neurotransmitter called dopamine. We've heard about dopamine before. And so depending on how dopamine is projected in that particular region, whether it binds to one particular cell type or another particular cell type, determines whether the brake or the gas are pressed. And in dystonia, the thought being that there's an imbalance in that, right? So there's excessive movement, and that balance between the brake and the gas are too much. And so this region of the brain, the substantia nigra, influences, it projects dopamine into this region, which is the basal ganglia. In fact, imaging, PET imaging, looking specifically at dopamine, so PET imaging being a component where we can inject radio tracers into the bloodstream, it goes into the brain, it crosses into the blood-brain barrier into the brain, and we can direct these tracers to bind to specific receptors, dopamine receptors in this instance. When it binds to those receptors, we can take a picture of the brain using PET imaging, and we can see exactly where those dopamine receptors or dopamine producing components are in the brain. And in fact, going back to this original CT, right, this is the stroke that caused dystonia. A little bit different slice. We're looking at the brain now, looking head on instead of from the top down. And in fact, that particular region where the stroke was seen that caused dystonia is a region of the brain where there is reduced dopamine. Cool, fantastic. We believe that dopamine has a role in this. We've been told that when we block dopamine in the brain via medications like Haldol, metoclopramide, etc., that people can get dystonia. This is all adding up. But unfortunately, this doesn't have a lot of translation. When we give dopamine, only in a very, very small subset of patients do they get better, right? And so, unfortunately, this has been a little bit of a dead end with respect to improving treatments. We had a lot of hope, we've had a lot of direction in this, but now what? What do we do now? And so we go back to the drawing board, and as complex scientists, we go back to complex models and imaging, and again, not going into the details of this, we recognize that there's another molecule, another neurotransmitter in the basal ganglia, acetylcholine, right, that influences how dopamine binds to that breaker, that gas pedal. And so this has now become a relevant region, a relevant neurotransmitter of study in dystonia, because we now have the tools that can do that. Just in the last 5 to 10 years, we've developed a radio tracer that can effectively bind in the brain to acetylcholine markers. And specifically in this case, it's called 18F-BAT, that doesn't matter. And in this upper picture, it's a beautiful picture, we see the brain in 3 different slices, sliced sideways, sliced in front, sliced on top. And this particular region, the basal ganglia, has a very high level of acetylcholine. And so the natural next question is, if acetylcholine influences dopamine, acetylcholine is high in this area that when we have a stroke or injury to it, it causes dystonia. Is there a difference of this level in patients with dystonia versus not? And that's what this bottom right slide shows. This is a preliminary data from our laboratory, but in fact, when we subtract a group average, healthy controls from dystonia, we see that acetylcholine levels in that particular area of the brain differ. Again, why does this matter? Because we now have particular drugs that may help patients with dystonia. This is still very early. We know, per one of the earlier slides, that anticholinergic drugs are the most effective at treating dystonia. The reason we don't use them frequently is because they also cause a lot of side effects. They cause blurred vision, difficulty thinking and memory, constipation, urinary symptoms. Patients don't tolerate these medications despite knowing that it works. That is because these drugs are very non-specific. Acetylcholine is everywhere in the body. You've heard about acetylcholine in the muscle. That's what Botox is inhibiting. That's not the acetylcholine I'm talking about. The signal of acetylcholine is very specific in different brain regions. It binds to different receptors, M1, M2, M3, M4. The current drugs affect all of those receptors throughout the entire body, hence these side effects. Recently, there have been new drugs that now are focused very specifically at some of those subtypes of receptors. And so the thought being that we can now focus these drugs to this particular region of the basal ganglia, to these specific receptors that may be dysfunctional. And so this provides some hope at this point that we may be able to develop medications. Again, very early in the studies. We still have a lot to go as far as proving this. There are some animal models that support this concept, but we have not hit clinical trials yet. But this is something that's on the forefront in the next few years with respect to what many groups in the country and the world are going to do. So I'm sorry I rushed through that. We were a little bit late to start with, but I think that hopefully I was able to convey some of the basic concepts that neuroimaging is key to understanding dystonia, right? And key to linking dysfunction of the brain causing dystonia. We've heard a lot of wonderful, very intelligent, smart, wonderful people who are treating dystonia by treating the peripheral manifestations, right? What's happening at the muscle level, what's happening at the nerve level, and how can we treat it? But if we can really tap into this brain dysfunction, this ideally is the best way to approach long-term treatment and hopefully a cure. Dystonia reflects network-level dysfunction, and this network-level dysfunction relates to biochemical dysfunction. And so linking that, using that knowledge of network-level dysfunction to tap into the specific chemicals that we might be able to treat with drugs is very important. We have tools, resting state MRI, that allows us to look at circuit-level dysfunction and find treatment potential with neuromodulation. So this gives us hope with respect to DBS and advancing DBS, TMS, other neuromodulation therapies. And then we have PET tools, which allow us to look at that molecular level and may give us hope with respect to new oral medications. So again, thanks to my group. This is our group at Washington University in the movement disorder section. Appreciate your time. I'm sorry we ran over, but I think maybe Dr. Holes, I can answer questions now, or maybe we bring everyone up and do a Q&A for everyone.
I think you start. Probably
perfect. So
you, and in the meantime, if I can get all the speakers to come up, we'll continue the Q&A session.
yeah, so yeah, I'll start. There's a question here.
Okay, I have MS, which is autoimmune disease. This is an autoimmune disease. D3, vitamin D3 is X for MS and for autoimmune disease, would that help with dystonias?
Yeah, so the question specifically was, would vitamin D3 potentially help with dystonia? And the context being that there's someone with a autoimmune disorder that responds or has found benefit with D3. There is no evidence currently that D3 would specifically help dystonia. There is in Dublin, Ireland, there was a reasonable consideration of autoimmune disorders potentially contributing to dystonia as well. That is to be determined and to be explored quite a bit. Some of those studies may be confounded by the fact that the immune system is in high, it's revved up high because we're injecting toxins in the body, Botox.